So welcome to the topic in animal biotechnology, right? We are still in the in vivo and in vitro models in animal biotech. So for today's topic, we are looking at neurodegenerative diseases. Okay, so we have the same learning outcome, which is to describe tools and techniques used in animal model systems, as well as applications and ethical concerns of animal biotech. So for today's topic, we are discussing on the in vivo models of human diseases, which includes um, the neurodegenerative disease. So as you can see here, neuro means the brain, okay, and degenerative here refers to the dying symptoms of the disease, meaning that the main part of the brain, okay, they are dying and results in the loss of the ability, for example, to speak, okay, or their intelligence level, the movement, their speech, right, is deteriorating. So in this topic, we are looking at two major diseases here. First is the Alzheimer's disease and also the Parkinson's disease. So this diagram here summarizes the overall view in terms of the types of diseases. We have Alzheimer's, the Parkinson's disease, and the amyloid, the symptom, as the Huntington's disease, the frontotemporal disease, and also the characteristics here. We have the Lewy body proteins here, the Lewy bodies, and also the prions are being indicated here. So we'll look into the pathology for the two main diseases, okay, and to investigate their pathology. So I have given a few examples here: Alzheimer's, Parkinson's, Huntington, amyotrophic lateral sclerosis, the ALS that I've mentioned earlier, and also the frontal temporal dementia. So these diseases are very, very diverse in terms of their pathophysiology, with some causing memory and cognitive impairments, and while others will affect the ability to move, speak, and also breathe. So I'll mainly refer to this journal here to develop my lecture notes. And after I have gone through my lecture notes, I will also go into this paper here to describe in detail what you would need to know. Okay, so we'll first understand the pathogenesis of Alzheimer's disease. They are characterized by early progressive memory loss and also followed by impairments in terms of the executive functions and behavioral disturbances, including agitation and paranoia. So we can see from here that there is degeneration in the neuronal cells within the brain. So as you can see here, there's a shrinkage of the hippocampus and also shrinkage of the cortex area in the Alzheimer's disease patients with enlarged ventricles. So if you go in depth into detail in terms of the pathology, you can see that they are accumulation of amyloid plaques and also tau tangles. So these two features are the main symptoms of the Alzheimer's disease, which results in damage and dying nerve cells. So Alzheimer's disease, they are characterized by three hallmark pathology. So the first one would be the amyloid plaques. We also call it the senile plaques, whose major insoluble components are the fibrillar aggregates of amyloid beta. This pathology is very specific to Alzheimer's disease. And the second one would be neurofibrillary tangles, right, which are characterized by tau proteins. So they are also found in numerous other neurodegenerative conditions besides Alzheimer's disease. And of course, these are also characterized by the hippocampal and cortical neurodegeneration. Okay, so if you look into the next slide, this really tells you what is it characterized by? The amyloid plaques as well as the tau tangles. So you can see here that there are numerous plaques or protein aggregates here where we call this as the amyloid plaques. So how are these formed is because of the cleavage of the amyloid precursor proteins. So these APP, amyloid precursor proteins, are the proteins, membrane-bound proteins here, where they are cleaved, meaning that they are cut and released into the surrounding. All right. So they are cleaved by the beta and gamma secretase, which leads to the formation of amyloid plaques in the intercellular space. So which means that if you can see from here, the abnormal function of the enzyme beta and gamma secretase, which results in the cleavage of the amyloid beta precursor proteins, which leads to accumulation of the amyloid plaques in the intercellular space. So you can see the structure of the amyloid fibrils here, okay, which leads to this accumulation. On the other hand, if you look at the neurofibrillary tangles, these are caused by misfolding of the proteins. Okay, so the tau proteins here are misfolded. So this leads to microtubule amyloid precursor protein and tau tangle formation. So as you can see here, it tangled up the neurites or neuronal cells projections here, which leads to neurodegeneration. All right. So for Alzheimer's disease, they are mainly characterized by these molecular characteristics in the form of amyloid plaques and also the tau fiber tangles. Right. So why is this happening? So research have found that they are mainly caused by mutations in the amyloid precursor protein, presenilin 1, presenilin 2, which results in autosomal dominant early onset of Alzheimer's disease. So there are a few models which mimic the Alzheimer's disease. So these are genetic models, meaning that they are um, working on the mutations of several genes that are associated with Alzheimer's disease, okay, hoping to recapitulate the symptoms and characteristics found in this disease. So we look at the two examples here. We have a genetic-based model of amyloid pathology. So we have talked about amyloid plaques earlier. So these models are looking at mutations in the amyloid precursor protein, the presenilin 1 and 2, in the transgenic mouse. So they are generating mutant mouse, transgenic rodents that contain the mutations in these few genes because these genes are mainly causing the Alzheimer's disease. So they are looking at overexpressing these genes, transgenic rodents, to mimic the amyloid pathology. The second model here, it looks at genetically based models of tau pathology, meaning that they are overexpressing these genes, okay, that cause the tau pathology. This also causes the FTDs, the frontotemporal dementia. Okay, so for models for the Alzheimer's disease, there are two types, those that mimic amyloid pathology and those that models tau pathology. All right. Okay, the next one we will be looking at Parkinson's disease. Okay, so for Parkinson's disease, they are characterized by the progressive loss of dopamine neurons. So dopamine is the main neurotransmitters that is responsible for the communications of neurons for signaling. So the loss of these neurons in substantia nigra pars compacta. This is a specific location within the brain. Okay, so if they have the progressive loss of neurons in this part of the brain, and they have the presence of misfolded alpha-synuclein in Lewy bodies and neurites throughout the nervous system. So these two are the main characteristics of Parkinson's disease. So they have the loss of neurons, with the loss of dopamine as all, and they will also have the presence of misfolded alpha-synuclein in Lewy bodies. So as you can see in this diagram here, understanding the pathogenesis of Parkinson's disease. So substantia nigra constitutes the part here, okay, the back part here. And in this area of the brain, they have a loss of dopamine neurons. So as you can see here, in the normal neurons that have the production of dopamines, which allow transmission of the neural signals. So in the Parkinson's disease, in the neurons, they do not have any transmission here, indicating that they have a loss of the dopamine-related neurons, which results in the loss of the ability for movement. So this will result in rigidity and slow movement and also tremors. So these are the key characteristics of Parkinson's disease. Okay, so what do the scientists understand about the pathogenesis of Parkinson's disease? They found that there are mutations in several genes that are related to Parkinson's. So these include alpha-synuclein, leucine repeat kinase 2, which causes autosomal dominant disease, while mutations in a few other genes, PINK1, DJ1, will cause autosomal recessive Parkinson's disease. So we have a few models for understanding this disease. We have the pharmacologic based model, which also means that they're using certain toxin or drugs to induce this disease. So historically, they have using these two uh, drugs here, the 6-OHDA and also the MPTP models. These are the two drugs which are used to induce Parkinson's disease, which also they are being successful in producing these symptomatic um, models. So we will look into this pharmacology model in detail in the journal paper later. And they will also have genetically based model for the alpha-synuclein pathology. So as you can understand in our previous slides, alpha-synuclein is the aggregates of the Lewy bodies within the neurons. So they are using genetic models of this alpha-synuclein gene, using a variety of different promoters that promote the mutant form expression of the alpha-synuclein. All right. And we have other genetic forms of the Parkinson disease model, which looks at different genes. So they have germline LRRK2 transgenic models, which is based on overexpression of the gene mutations, which shows different degrees of the dopaminogenic abnormalities. And we also have the seeded models. So what are the seeded models here is that they are not they are not using any drugs to induce. They are not using any genetic mutations or transgenic rodents. What they are doing here is they inoculate, meaning that they are doing the intracerebral injection of purified aggregates, the proteins, the disease lysates of the or the mouse brain lysates directly into the mouse, the model. So they are hoping that by directly injecting these protein aggregates into the brain of the mouse, it will replicate the similar pathologies and symptoms in the mouse to reflect the Parkinson's disease. So we looked into their phenotypes in the journal later as well. So these are called the seeded models. So what are the common challenges in modeling the neurodegenerative disorders in rodents? Is that mouse or rats that have short lifespan, okay, which may contribute to incomplete development of the pathological hallmarks or neurodegeneration. So As you can see, human they have an average lifespan of 60 to 70 years, while for rodents they are on average 2 to 3 years. So the difference in terms of the lifespan may also contribute to the hallmarks and the neurodegeneration. And of course, there are inherent differences in the development and function of the rodent and human brains. So we have different in terms of the anatomy and physiology of the brain in the rodent and human brain. They are, of course, very different. So which may also contribute to one of the challenges in modeling neurodegenerative disorders in mouse. And there might be some limited relevance in terms of the very rare genetic variants for understanding and treating the sporadic disease. So some patients they might have different genetic causes, okay, which are rare differences in terms of the genes that causes the mutations, which is very difficult to be replicated in the mouse. Of course, the genome differences between rodent and human may have profound implications when modeling the disease, okay? And there are inherent caveats related to methods used to generate these animal models. So we will look at one of the paper later on in terms of a slight differences in their experimental methods of analyzing cognitive behavior in mouse would really generate different results. Okay, so we'll look at these differences in terms of the methods used for analyzing results of cognitive behavior and also the limitations related to the use of inbred animals that do not reflect genetic diversity of the population. So when we are testing the similarities in terms of animal models and human disease. And most of the animals that we are using, they are inbred animals, meaning that they have limited genetic diversity, which is very different to the overall human population and the the in terms of the mechanism that it will bring in terms of the diversity from the human population. So we will also look at some non-rodent models, like for example, Drosophila, C. elegans, yeast models, or even the zebrafish. They are also used extensively to understand neurodegenerative disease that contributes to the pathology. So I have one question here. Okay, so what are the approaches to translatability of neurodegenerative animal models? Meaning that how can we improve, okay, to use animal models to reflect the understanding and improve the how we can better model the characteristics that we see in human pathology and the one that we see in the animal models.